So today we are going to discuss hemoglobin, myoglobin, and some diseases. Okay, so let's start. So what is hemoglobin? হেমোগ্লোবিন কি আছে বলো, কে বলবে? আয়রন কন্টেনিং প্রোটিন, আচ্ছা, প্রেজেন্ট ইন where? RBC. Okay, one RBC, can anyone tell me how much molecules of hemoglobin present? One RBC, how many hemoglobin molecules present? কে বলতে পারবে? একটা আরবিসিতে কতগুলো হিমোগ্লোবিন মলিকিউল থাকে? এনি আইডিয়া? নিয়ার অ্যাবাউট 300 মিলিয়ন, রিমেম্বার। ওকে, and one hemoglobin molecule carries 4 molecule of oxygen. Okay, so one RBC can carry up to 1.2 billion of oxygen molecule. Understand? I am coming later. Just listen. So today our discussion topics. This is the competency 5.9. Hemoglobin, myoglobin, types of hemoglobin, derivatives of hemoglobin, variants of hemoglobin, and physical and pathological elements we are going to discuss today. So what is myoglobin? This is one type of hemoglobin, but difference from hemoglobin is it only one single monomer, but hemoglobin has 4 monomers, tetramers hemoglobin. It is present in cardiac cell and skeletal muscle. Okay, function is to oxygenation, carry oxygen. So this is myoglobin. This is monomeric oxygen binding hemoprotein found in heart and skeletal muscle. It is a single polypeptide chain with heme moiety, molecular weight 17,000, and structurally resembles is subunit of hemoglobin molecule, and function is to reserve oxygen. Okay, it further serves as oxygen carrier that promotes the transport of oxygen to the rapidly respiratory cells, muscle cells. In case of injury, muscle injury or cardiac injury, myoglobin come into circulation. Okay, but it is not specific. Myoglobin is an early marker of diagnosis of myocardial infarction, but this is not specific. Okay, not for cardiac muscle, it is present also in skeletal muscle. This is the reason it is not specific. Okay, but in case of muscle injury or crush injury, massive amount of myoglobin can come into circulation, and via circulation it reaches into kidney, where it passes through urine called myoglobinuria. Actually this is toxic material. It can damages renal tubule cells, myoglobinuria. Okay, in case of multiple crush injury like road traffic accident, patient may develop myoglobinuria, and this myoglobin can causes tubular necrosis. Understand? Renal tubular necrosis ultimately causes acute renal failure. This is the importance. Understand? You will study further in your future lecture classes when you are going to teach surgery, pathology. Understand? Okay, so this is about something about myoglobin. So you may ask in the exam, write down myoglobin is a short note. Okay, myoglobin short note. So even if you ask myoglobin, then these points should be noted. in your exam paper. Okay, this is something about myoglobin. Okay, let's start about hemoglobin, very important topic. Okay, hemoglobin, it is only present in RBC. Okay, and this is a tetrameric, tetra means 4, tetrameric metalloprotein, because in hemoglobin there is iron molecule, is iron present, Fe plus 2. That is why it is called metalloprotein. It transports oxygen to the tissue and returns carbon dioxide and proton to the lung. One molecule of hemoglobin can bind up to 4 molecules of oxygen. And one RBC contains about 300 million of hemoglobin molecules. Hemoglobin has the buffering function within the RBC. As you learned already in your acid-base buffering system, you know that hemoglobin also has some buffering capacity. What is buffer? Remember anyone? Buffer ki bolte hobe? What is buffer? Buffer is a solution which resists change of pH. Example of buffer. One example, hemoglobin buffer. Apart from this, phosphate buffer, bicarbonate buffer. Very good. Okay. Okay. To ensure adequate tissue oxygenation, you must have sufficient hemoglobin level in your blood. And when we going to measure hemoglobin, we express it as gram per deciliter. Okay. What is the normal value of hemoglobin in case of male and in case of female? Male 13 to 18 gram per deciliter. Okay, broadly remember this, 13 to 18 gram per deciliter. In case of female, 12 to 16 gram per deciliter. But unfortunately, in our India, majority of ladies which are in reproductive age group suffer from anemia. They have hemoglobin level less than 10 gram %. 10 gram per dm. Okay, so this is very common in rural India, rural area. We found children suffering from hemoglobin anemia because of worm infestation. Worm. Understand? This is the reason Government of India started deworming campaign in each school. Government giving free of cost anti-worming drug like albendazole. Okay. So hemoglobin, its primary function to transport oxygen from lung to tissue, and from tissue it carries carbon dioxide and some proton to the lungs. Okay. Hemoglobin is an important buffering agent. One RBC molecule present near about 300 million of hemoglobin. Okay. So, and one hemoglobin molecule carry 4 oxygen molecule. So, if you calculate, one RBC molecule can carry 1.2 billion oxygen molecule. Okay. You may ask in the exam, Viva. Remember, this is extra knowledge. One RBC has 300 million of hemoglobin molecule. Okay, let's start. Let's move. Okay, Bohr effect. We already read in physiology lecture classes probably. What is Bohr effect? The binding of oxygen to hemoglobin decreases with increasing H+ concentration, that is lowering pH, or when the hemoglobin is exposed to increased carbon dioxide. This phenomena is known as Bohr effect. This is due to change in binding affinity oxygen to the hemoglobin. Bohr effect causes shift oxygen dissociation curve to the right. You can see here This is pH 7.6, pH 7.2. Okay, so pH is decreases and this curve shifted towards right side. This is called Bohr effect. Bohr effect is primarily responsible for the release of oxygen from the hemoglobin to the tissue. This is because of increasing partial pressure of carbon dioxide and decrease in pH in the actively metabolizing cell. Bohr effect. So we already learned in physiology chapter classes what is Bohr effect. This is a brief discussion. Bohr effect in biochemistry. Effect of 2,3-bisphosphoglycerate on a oxygen affinity of hemoglobin. 2,3-bisphosphoglycerate or 2,3-BPG is the most abundant organic phosphate in the erythrocyte. Its molar concentration is equal to that of hemoglobin. One hemoglobin molecule is 1,2,3-BPG. So, if we have 100 number of hemoglobin molecule, that means we have 100 number of 2,3-BPG molecule. Okay. It is produced in the erythrocyte from the intermediate of glycolysis. There is 1,3-bisphosphoglycerate. This short pathway referred to as Rappaport Liebering shunt. You may ask in the exam most probably short note about Rappaport Liebering shunt. You asked, no? So this is the Rappaport Liebering shunt. During this shunt, 2,3-DPG generated from 1,3-BPG. Binding oxygen depend on pH, okay. Partial pressure of carbon dioxide. We already read in our acid-based lecture classes, and also depend on 2,3-BPG, bisphosphoglycerate. Okay. Next. So you can see this is a normal blood with 2,3-BPG. Here, in patient of anemia, there is less hemoglobin, so there is less BPG. So this curve shifted towards right side. And steep hemoglobin, not 2,3-BPG, and shifted towards left side. There is no 2,3-BPG, so it is shifted towards left side. This diagram I taken from your favorite book, Satyanarayan. You can read for Satyanarayan. Okay, next. Hemoglobin structure. This is a complex thing. I am giving you some shorter thing. So you can see hemoglobin has tetrameric. There is 4 globin chain, alpha 1, alpha 2, beta 1, beta 2 globin chain. Okay. In the center of the globin chain, there is presence of heme protein. Okay, and hemoglobin is a globular structure. Here, the globin chains. The outside we have hydrophilicity outside, but inside hydrophobicity. In the hydrophobic center, heme protein is present. You can see here, this is the 3-dimensional structure of hemoglobin. Here we have alpha 1, alpha 2 chain, beta 1, beta 2 chain. Here this is the 2,3-BPG molecule in the center, and in the center of each globin chain we have heme molecule 1, 2, 3, 4. This is 2,3-BPG. Okay. So you can see structure of adult hemoglobin. It has 4 subunit, alpha 2 alpha chain and 2 beta chain. Has relatively hydrophilic surface and hydrophobic interior. Each subunit has a heme group within a hydrophobic pocket. It has alpha beta 1 dimer and alpha beta 2 dimer. Understand? So hemoglobin is 3-dimensional structure. Everyone make a imagine inside your brain. What is the structure of hemoglobin? It is a globular structure, round like shape, and It also has 4 globin chain. Okay, at the outside there is hydrophilic amino acids are there, and inside there is hydrophobic is there, and inside each globin chain there is heme protein is there, and inside this hemoglobin there is 2,3 BBG is there. Okay, so this is the deoxygenated form of hemoglobin. Next is developmental variations hemoglobin molecule. This is very important. Why? I am telling you later. Almost all types of hemoglobin from fetal life to adult life are made up of alpha chain and 2 alpha chain and 2 non-alpha chain, either beta, gamma or delta. Cytosynthesis is starting from 15 days of fertilization up to 6 weeks, which is from Biochemistry. And in the liver after 6 weeks until the 30 weeks prenatal life. Okay, and next after 30 weeks it is going in bone marrow and some flat roots in the skull. Okay, this is the hemoglobin development. First 15 days, starting from 15 days up to 6 weeks in yolk sac. After 6 weeks to 30 weeks in the liver, and then after 30 weeks from bone marrow. Usually long bone and some flat bone in the skull. Okay, face. So you can see development variation hemoglobin, subunit combination, embryonic hemoglobin. Here you can see after 15 days and after 6 weeks 50% alpha chains achieved, and it almost throughout the life maintains 50% of alpha chain, and after a few days, say few weeks, gamma chain developed, and next 30 weeks it start to decline. After birth it declined sharply, and after 18 weeks and 30 weeks is after birth is achieving in adult value, and the rest of the life it maintain 1 to 2%. Okay, this is gamma chain, and also you can see here some other chains also going to develop and disappear after 6 weeks very rapidly, like epsilon chain or sigma chain, zeta chain. Okay, this is also important. Just see, make a imagination. If you don't know this, no problem. This is one extra information. Fetal hemoglobin HbF is made of 2 alpha chain and 2 gamma chain. 1% of total blood hemoglobin level. In adult hemoglobin HbA, 97% of total hemoglobin level. A minor adult hemoglobin like HbA2, 2% of total hemoglobin level. Okay. Do you understand, everyone? No confusion? Understand? বুঝতে পেরেছো? বুঝতে পারোনি? আচ্ছা. So this is a graphical representation of alpha chain development. Okay. After few weeks, say 15 days, alpha chain start to develop and achieve is almost 50% value after 6 weeks, and throughout the life it maintained. Understand? And after few weeks, here gamma chain develop and achieves after 18 weeks is achieves near about 50% value. Okay, so this is the prenatal age. This is birth period and this is postnatal age. So after 18 weeks we have hemoglobin F, that is consists of 2 alpha chain and 2 gamma chain. So in prenatal age we have predominantly HbF, fetal hemoglobin. Okay, but after 30 weeks it start to decline, and after birth it rapidly decline this gamma chain and reaches up to 1 to 2%. And here you can see after 6 weeks beta chain start to develop, and after 30 weeks it rapidly increasing its value, and after 6 to 18 weeks is almost 50% value achieved, and it forms hemoglobin A. Adult hemoglobin consists of 2 alpha chain and 2 beta chain and reaches its adult value. Now understand? Okay. Achha, you can see here, apart from alpha chain and gamma chain and beta chain, we have 2 other chains. This will epsilon chain, another chain is zeta chain is there. I am coming later. And they can make some minor amount of hemoglobin. What is that? I am coming later. Just see this. So types of hemoglobin in adult, as I told earlier, you can make a screenshot. Adult hemoglobin. 97% hemoglobin, adult hemoglobin consists of 2 alpha chain and 2 beta chain. Okay. And some other variants, minor variants are there. This for HbA2, 2%, 2 alpha chain and 2 delta chain. And HbF, there is 2 alpha chain and 2 gamma chain, and 2 alpha chain 141 amino acid residue. It is formed from chromosome number 16. And beta chain, gamma chain, delta chain, there is 146 amino acid residue that is derived from chromosome 11. Okay. So this is very important. You may ask MCQ. Understand? Very important information. This is the adult hemoglobin. Okay. बुझते पेरेছো সবাই? समझ गए आप समझ गए? हाँ, खड़े हो जाओ. बोलो. What is adult hemoglobin? इसमें कितना रहता है? अल्फा चेन कितना रहता है? कितना बीटा चेन रहता है? ये क्या है? HbA. दिस इज कॉल्ड एडल्ट हेमोग्लोबिन. समझे? हेमोग्लोबिन क्या होता है? उसमें 4 तो ग्लोबिन चेन रहता है. 2 अल्फा रहेगा, इनको से एडल्ट हेमोग्लोबिन 2 अल्फा चेन रहेगा और 2 बीटा चेन रहेगा. समझे? इनकेस ऑफ एडल्ट हेमोग्लोबिन दिस इज 97%. इन ए नॉर्मल पर्सन, से यू आर नॉर्मल पर्सन, if you going to check your hemoglobin status, how can you check? By electrophoresis. Then you can see we have 97% adult hemoglobin. Near about 97%, very actually. It has 2 alpha chain and 2 beta chain, gamma globin chain. Okay. Apart from this HbA, we have also some other variant, minor variant like HbA2. There is called. It has consisted of 2 alpha chain and 2 delta chain. समझे? And apart from this, we have very minor amount, say 1%, less than 1%. This is fetal hemoglobin. हाँ? इसमें 2 alpha chain रहेगा और 2 gamma chain रहेगा. ठीक है? In case of children, say हाँ. So you can see in this graph. देखो इसमें ग्राफ में देखो, ये क्या समझे ग्राफ देख के? बताओ तो. तो समझे कुछ भी? हाँ बोलो, जोर से बोलो. हाँ बोलो. वेरी गुड. हाँ. ठीक है, चलो बैठ जाओ. ओके. तो प्लीज़ लिसन किया है फुली, है ना? ऐसा नहीं. नेक्स्ट, अदर हेमोग्लोबिन वेरिएंट्स. In case of adult people suffering from diabetes, nowadays we going to test HbA1c. You've heard the term HbA1c. This is one type of variant of adult hemoglobin. Please remember, HbA, not A2, not F, only HbA. Okay. This is because non-enzymatic glycation of hemoglobin with glucose in the N-terminal valine of beta chain in hemoglobin. Can you understand this term? HbA में 2 अल्फा चेन रहता है, 2 बीटा चेन रहता है. ठीक है। बीटा चेन का जो एन टर्मिनल होता है ना, इस पेप्टाइड चेन से एन टर्मिनल, कार्बोक्सिल टर्मिनल, in n terminal we have valent residue. In this valent residue जो एक्स्ट्रा शुगर रहता है, शुगर come and attached to it, it form a glycation, that is called glycated hemoglobin. This is non-enzymatic. Once it formed, it cannot revert back. So the hemoglobin is formed, glycated hemoglobin, it cannot be converted into normal hemoglobin. Please remember, it remain as it is up to the whole life of the RBC. Okay, what is the lifetime of RBC? 120 days. Very good. So that means half life 60 days. Okay. So in case of patient suffering from diabetes mellitus, we can see the percentage of glycated hemoglobin. In normal people, say I am a non-diabetic person, if I am going to test HbA1c level, I also have some glycated hemoglobin because I also have sugar, but I am non-diabetic, so I have a limited number of HbA1c, so less than 6% normally. But in case of diabetes patient, say he is suffering from high blood sugar, so he also increases the level of percentage of HbA1c. So this is only in adult hemoglobin. Please remember, if you going to test one baby, say newborn baby suffering from diabetes, you are going to see HbA1c level value. Now can you tell me, we are going to find HbA1c level or not? In case of newborn baby, where adult hemoglobin is very less, it is more than 50% fetal hemoglobin, you cannot get HbA1c. Okay? I have seen some mistakes done by some of the doctors who prescribe HbA1c test in case of newborn baby. This is absolutely wrong. Understand? So HbA1c is a isoform of hemoglobin A, adult hemoglobin, not other, because we should have beta chain. This only occurs in beta terminal of n terminal valent of beta chain. Okay, you understand? HbA1c किससे बोले? Normal value less than 6%. Okay, this is very important. HbA1c. Apart from some other variants there, you don't have to remember like HbA1a, HbA1b, HbA1c. You don't have to remember everything. Just remember HbA1c. Okay. Let's move forward. So this is embryonic hemoglobin. What is embryonic hemoglobin? If hemoglobin made up of 2 zeta chain and 2 epsilon chain. Here you can see epsilon chain and zeta chain. Is these 2 chain make a hemoglobin that is called hemoglobin Gawar 1. Hemoglobin Gawar 1. Okay. 2 epsilon chain and 2 zeta film, they form hemoglobin Gawar 1. This is very short period of time. After that, it will disappear. Only in fetal life. Next is hemoglobin Gawar 2. Here it is formed by 2 alpha chain and 2 epsilon chain. Here you can see 2 alpha chain and 2 epsilon chain. You can make hemoglobin Gawar 2. And next is hemoglobin Portland. Here it made by 2 zeta chain and 2 gamma chain. 2 zeta chain and 2 gamma chain, it make hemoglobin Portland. It is only present in fetals. Okay. As soon as you start making hemoglobin from liver, all the things disappear. So if you ask in the exam, please tell me some fetal hemoglobin. Can anyone tell me? Can you answer na? You can answer तो समझ गए ना? अगर एग्जाम में हम लोग पूछेंगे, बोलो 2-3 हीमोग्लोबिन जो बच्चों में मिलता है, बच्चों में नहीं फीटस में मिलता है, तो आपका आंसर दे सकते हो ना? Gawar 1, so this is the answer. HB Gawar 1, HB Gawar 2 and hemoglobin Portland. Okay. There are some variants of hemoglobin, types of hemoglobin. Any question till now? No question, no? Let's move. So already discussed this graph before. This is the chromosome 1611. You don't have to remember anything. This is only for PG purpose. Right now just focus on basic things. Next is fetal hemoglobin A2, HbA2. This normal hemoglobin, 2% hemoglobin, 2 alpha chain and 2 delta chain is present in adult people. HbA2 made up by 2 alpha chain and 2 delta chain. Delta chain has sequence homology with beta chain. In beta thalassemia, what is beta thalassemia? I am coming later, just remember. Beta thalassemia as a compensation of HbA2 is increased. Thalassemia, have you heard the term thalassemia? Thalassemia is a blood disorder. There, a deficiency of the chain, the nomenclature given. Say example, we have beta chain is absent or deficient. This is called beta thalassemia. In case of alpha chain is absent or deficient, this is called alpha thalassemia. Okay. In case of beta thalassemia, there is beta chain is absent, so in compensation HbA2 level is increased. Okay. If you are going to study electrophoresis hemoglobin, then in case of thalassemia patient, you can see A2 level is increased. I have seen. When I work in AIMS, they regularly do hemoglobin electrophoresis, so I can study this. Okay. These are not magic, these are all science actually. This is really happen. Fetus hemoglobin, HbF has a 2 alpha chain and 2 gamma chain as I told earlier. Main difference in compared to HbS are deoxy, HbF is more soluble and slower electrophoresis mobility for HbF. HbF is more resistant to alkali denaturation and it is decreased interaction with 2,3-DPG. This is very important line. HbF has decreased interaction with 2,3-bisphosphoglycerate. As a result, what happened? This increase in oxygen affinity is physiologically advantageous in facilitating transplacental oxygen transport. Okay. So you know, fetus receive oxygen from mother blood. Okay. So if the fetus has more affinity to the oxygen, that means they have more amount of oxygen present in their blood. This is done by HbF. Okay. So you may ask in the exam reasoning. Fetus hemoglobin carry more oxygen in their blood. Now you can answer, because in fetus hemoglobin we have more amount of HbF and each HbF has less number of 2,3-DPG, so it has more affinity to the oxygen. So in case of normal oxygen percentage, say 20% we are going to supply. So in fetus, it can have 50% of oxygen. Okay. When percentage of oxygen is 20 mmHg, the fetus is 30% saturated. This is because of this thing. Decreased interaction with 2,3-DPG. Okay. Synthesis of HbF start by 7th weeks of gestation. It becomes the predominant HbF by 22 weeks of life. At birth, 80% hemoglobin is HbF. During the first 6 months of life, it decreases to about 5% of total. When HbF level may remain elevated in children with anemia and beta thalassemia as a compensatory mechanism. This is already described from this topic. Now some diseases hemoglobin. Most important two diseases, sickle cell disease and thalassemia. Let's discuss one by one in short. For a sickle cell disease, for sickle cell anemia, also called HBS, is the most common form of abnormal hemoglobin. It is very common. It is so named because the erythrocytes of these patients adopt a sickle shape. What is sickle? Means crescent shape. Crescent shape. Half moon like shape. Crescent. Okay? Sickle. Okay? So in low oxygen concentration, the RBC take a shape like crescent. Sickle. This is called crescent shape or sickle shape. Normally RBC shapes like this, biconcave shape. Okay, this is normal. This is sickle. So what happened when RBC molecule try to escape in the splenic sinusoid? They trap in the splenic sinusoid and they destroyed by the sinusoidal cells, macrophages. Okay. So patient is suffering from sickle cell disease, sickle cell anemia. They have massive splenomegaly and anemia. This is because of HBS. Molecular basis of HBS. You may ask in the exam, please remember this is a very important question. Short note, you may ask sickle cell anemia. If you write this thing, molecular basis HBS, in case of sickle cell anemia, the hemoglobin S, it is a two normal alpha chain and two abnormal mutant beta chain, globin chain. This is due to difference in the single amino acid in HBS. Glutamate at 6th position of beta chain is replaced by valine. This is very important. Almost 1 or 2 years PG question come from this. Okay. And you can see here, this is bicambal shade, but this is sickle cell. So malaria parasite cannot remain inside this cell. It will die. So they have the patient suffering from sickle cell anemia. They have some resistance against malaria infection. Okay. Yes, because in low oxygen, the RBC take a shape like this, and inside this RBC, malaria parasite cannot survive. বুঝতে পারলে? Okay. So if you ask an exam sickle cell anemia, you must write this, at least this line. HBS glutamate is 6th position of beta chain is replaced by valine. Okay. Next, how can diagnose sickle cell anemia? This is a sickling test. This is a simple microscopic examination of blood smear prepared by adding reducing agents like sodium metabisulfite. Sickled erythrocytes can be detected under the microscope. This is sickling test. Apart from this, we can go for electrophoresis. In electrophoresis, the slow mobility of HBS is due to less negative charge caused by absence of glutamate residue that carry negative charge. In case of sickle cell trait, the fast moving HBA and slow moving HBS are observed. Okay. Treatment. This is genetic disease, so there is no treatment, just symptomatic treatment, blood transfusion. Okay. Next, thalassemia. Thalassemia, as I told earlier, this is absent of one or more than one chain. According to the chain deficiency, the nomenclature given. Say one alpha chain deficiency, this is alpha thalassemia. Beta chain absent, this is beta thalassemia. Okay. In case of alpha thalassemia, this is caused by decreased synthesis of or total absence of alpha globin chain of hemoglobin. There are 4 copies of alpha globin gene, 2 on each one of the chromosome number 16. 4 types of alpha thalassemia occur, which depend on number of missing alpha globin gene. Okay. This is taken from your book Satyanarayan. This is normal thing. Here we have 2 alpha chain, alpha 1, alpha 2, beta, and this is from mother, this is from father. Okay. Clinical symptom now. In silent carrier, one chain is, so this is mutant or absent. Till this is no symptom because of compensation. Here in case of trait, here from mother and father absent. Minor anemia is there. In case of hemoglobin H disease, here alpha 1, alpha 3 is absent. Alpha 1 both is absent, but alpha 2 is there. Here mild to moderate anemia may lead to normal life. Okay, trait and HB almost same thing. And if in case of alpha 1, alpha 2 both are absent, then fetus will die within the womb. This is called hydrofetalis. Okay. This is something about alpha thalassemia. You will read again in your pathology classes, medicine classes later. Thalassemia, just heard the term alpha thalassemia. Next, beta thalassemia. This is decreased synthesis or total lack of formation of beta globin chain causes beta thalassemia. Here, beta thalassemia minor or beta thalassemia major. All of you heard the name अमिताभ बच्चन, नाम सुना होगा आप लोगों ने. अमिताभ बच्चन का उनका थैलासेमिया ट्रेट है, लेकिन उनका जो पत्नी है जया बच्चन वो तो नॉर्मल है, इसलिए उनका 2 बच्चा सही है, अभिषेक बच्चन और श्वेता बच्चन. ओके, ही इज ए फेमस पर्सनैलिटी. थैलासेमिया ट्रेट अमिताभ बच्चन. ओके. In beta thalassemia minor, one chain is deficient or absent, but another chain is there, so compensation is there. Patient has some minor anemia. But in case of beta thalassemia major, both the beta chains are absent, so patient develop as the child started going after birth, when HbF level started to decline and supposed to happen fetal hemoglobin. Okay, so baby develop anemia with splenomegaly, and baby have to go for repeated blood transfusion. Otherwise, baby cannot survive. This is very common beta thalassemia. Okay, this is the reason nowadays before marriage couple go for thalassemia screening test. Okay, thalassemia. Here you can see beta thalassemia. 2 beta chain are not present. This is normal. One chain is absent, so this is thalassemia minor. Both chain is absent, so this is thalassemia major. Okay. Treatment: repeated blood transfusion, symptom treatment, and bone marrow transplantation. Nowadays happen bone marrow transplantation. Other form of hemoglobin, HbC, hemoglobin C disease, Colles hemoglobin HbC is characterized by substitution of glutamate by lysine in the 6th position of beta chain. Due to presence of lysine, HbC moves more slowly on necroporosis, compared to HbA and HbS. This disease is characterized by mild hemolytic anemia. No specific therapy is recommended. Hemoglobin D disease. This is caused by suppression of glutamate in place of glutamate. Glutamine into glutamate in 125th position of beta chain. Several variants of HBD are identified from different places indicated by this suffix. For instance, HBD, this is Punjab, HBD is Los Angeles, like this. Okay. HBD on electrophoresis move along with HBS. You will be surprised to see when you go for hemoglobin electrophoresis, there is more than thousands number of hemoglobin variants are there. So it is not possible to remember everything. Okay. So remember this thing: HBC, HBD, HBS. Okay. This is HBE. This is also common. HBS, and this is common in this area, northeast region, Bangladesh, West Bengal. Okay. In Southeast Asia. HBE is characterized by replacement of glutamate by lysine at 26th position of beta chain. In individual of HBE, either homology or heterology have no clinical manifestation. This is fortunately there is no clinical manifestation. Okay. So these are something about hemoglobin and their variant. Please go for this classes after completion lecture. Very important topic. You may ask in the exam 1 or 2 questions. Thalassemia, sickle cell disease, hemoglobin structure, myoglobin. Okay, these are very important. Sickle cell disease. Okay. Types of hemoglobin. English of adult, English of fetus like Portland B hemoglobin, GAWAR 1, GAWAR 2. Okay, this is hemoglobin. Okay, any question? What is the structure of hemoglobin? Bolo. Globin kota globin thake. Kota chain thake. How many chains are there in hemoglobin? Charte thake, charte. It is called tetrameric structure. Okay. দেখতে কি রকম হয়? How it looks hemoglobin? Is it elongated or globular or circular? What? Circular, globular structure. Okay. Inside the globin, we have another 4 globin chains. Okay. Alpha 1, alpha 2, beta 1, beta 2. They form tetrameric structure. Okay. Outside there is hydrophilic nature. That means they can attach with water. Okay. And inside there, hydrophobicity is there. They hate water. And in the inside of each globin chain, we have heme molecule. Okay, what is heme molecule? There is a iron containing molecule. This is heme protein. Heme protein. It is called pyrrole ring. Heme pyrrole ring. Inside there is Fe+2 is there. Okay. So this is one alpha chain. This is one alpha 2. This is one beta 1, beta 2 chain, and this is 2,3-BPG, bisphosphoglycerate, and this is the heme molecule. You can see heme molecule. Okay, this is the short form heme molecule. One molecule, alpha 1, alpha 2, beta 1, beta 2. So this form a globular structure. Please tell me something about thalassemia. According to the absences of chain, the nomenclature is given. Say alpha chain is absent, there is alpha thalassemia. Beta chain is absent, this is beta thalassemia. Okay. What is the clinical manifestation? Beta thalassemia is very common. Okay. So when I work in West Bengal, I am seeing at least daily in our medical college, Bardhaman Medical College, 50 to 60 children come for blood transfusion. Okay. So in beta thalassemia major cases, we have to give blood transfusion repeatedly. Okay. All of you know blood is iron. So eventually the iron start to accumulate inside the body and baby will die because of iron overload. This is very unfortunate thing. This is called hemochromatosis, iron overload. So sometimes we have to give chelating agent like desferrioxamine to treat their iron overload.